Alrighty. Covalently bonded compounds will separate into component parts when placed in water. True or false? Okay. When ionically bonded substances are placed in water, the slightly negative positive ends of the water molecules cause the ions to leave each other and interact with water molecules instead. So ionic bonded, not covalent bond. This is ionic bonded substances that do this. In this way, the polarity of water dissociates salts in any water solution, such as the internal environment. Because the resulting solution contains electrically charged particles, ions, it will conduct an electric current. Substances that release ions in water are therefore called electrolytes. So they're ionic bonds, not covalent ones. Two or more atoms that bond together form a blank isotope, ion, element, or molecule. Two or more atoms that bond together are called what?
Would it be a molecule?
They're molecules, right? Good job. Compounds that are compounds are classified as acids if they release blank when they dissociate in water. Compounds are classified as acids if they release what? Hydrogen ions, hydroxide ions, salts, or bases. When ionically bonded substances are placed in water, the slightly negative and positive ends of the water molecules cause the ions to leave each other and dissociate. For example, the salt sodium chloride releases sodium ions and chloride ions when it is placed in water. In this way, the polarity of water dissociates salts such as the internal environment. Okay, so these are electrolytes. We need to scroll. This doesn't tell us about acids. This was the previous question. Acids are electrolytes that release hydrogen ions in water. So acids are electrolytes that release hydrogen ions in water. So hydrogen ions is what you want to use there. All right. What two types of information can be determined from the molecular formula of a molecule? How the atoms of the molecule are arranged in space, the atomic weight of the molecule, the types of atoms in the molecule, the numbers of atoms in the molecule. Two of those answers are correct. the molecular formula of a molecule.
Would it be the number of atoms in the
I
molecule?
believe so. There's two answers that they want. You can look it up if you. Okay, a molecular formula represents the number and types of atoms in a molecule. So the number of atoms in a molecule and the type of atoms in a molecule. Such a formula displays the symbols for the elements in the molecule and the number of atoms of each element. For example, the molecular formula for water is H2O, which means that each water molecule consists of 2 atoms of hydrogen and 1 atom of oxygen. The molecular formula for glucose is C6H12O6, indicating that each glucose molecule contains 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. So it's the number of atoms in the molecule and the type of atoms in the molecule. You already selected the number, so the other one is the type. Good job. Nice. Okay. Bases are electrolytes. Bases release ions in water that bond hydrogens, or bases release hydrogens in water. What's true about bases? Okay, so here we have, okay, that's the first part, that they have to be ionically bonded. Acids we read about before, they release hydrogen ions. Then we have the pH scale, and we're looking for basic. So we want something about bases, right? Okay, that's buffers, that's bases. You can look at the answer if you want. Acids are electrolytes that release hydrogen ions in water and have a pH less than 7. Bases are electrolytes that release ions that can bind to hydrogen ions in water. So they bind, they release ions that bind to hydrogen. Go up a little bit. No, up. Like back toward the top. I'm looking for the answer to what a base is. How does the hydrogen ion? Okay. Okay, that's your concentration. That comes later. Okay. Let me see. pH the midpoint, said to be neutral. Okay. Okay, I think it's in the paragraph before this, where they were talking about, yeah. So here it says the hydroxide ions in turn can bond with hydrogen to form water. Thus, sodium hydroxide is a base. So here's the second, you see the second yellow part? So acids are electrolytes that release hydrogen, and electrolytes that release ions that bond with hydrogen are bases. So if you release the hydrogen ions themselves, then you're an acid. If you release the ions that bond with the hydrogen, then you're a base. Okay, so let's see what the question says. So it would be the second one. Release ions in water that bond hydrogen. Because if you released hydrogen, you'd be an acid. I think that's what they want. They want two? Oh, and they wanted the other answer. They were electrolytes. So the answer we picked was right, but we needed two answers for this. They're also considered electrolytes. So they're electrolytes that release ions in water that bond hydrogens. So they wanted both of those. Okay, so we got a half right. A product is a molecule that is combined with another molecule in a reaction, the components used for synthesis, or a more complex structure made from reactants. So what is the definition of a product here? Okay, so chemical reactions form or break bonds between atoms, ions, or molecules, generating new chemical combinations. For example, when two or more atoms, reactants, bond to form a more complex structure called a product, the reaction is a synthesis. Okay, so the product, so you have A is a reactant, B is a reactant, and AB would be the product. So the more complex structure that reactants form is a product. Mm-hmm. And so we're looking for the definition of a product. It's in the yellow. I just read it to you. Two or more atoms, reactants, bond to form a more complex structure called a product. So a product is a more complex structure that is made from reactants. And it's also yeah, okay. I think that's correct, yeah. If a compound contains carbon and hydrogen together, it is called a what? Molecule. Carbon and hydrogen together. The book will tell us. Chemicals, including those that enter metabolic reactions or are produced by them, can be divided into two large groups. Compounds that include hydrogen, carbon and hydrogen are called organic. So if they have carbon and hydrogen, they're organic compound. Mm-hmm. Nice. Okay. What type of bonding is occurring between the water molecules in this figure? Hydrogen bonding, ionic bonding, or nonpolar covalent, between the water molecules, not within the water, but between them. Within the water, between the water molecules, what do we have? Okay. Okay, so remember we talked about weak hydrogen bonds. Typically polar bonds, your screen shrank. Typically polar covalent bonds form where hydrogen atoms bond to oxygen or nitrogen atoms. The attraction of positive hydrogen end of one polar molecule to the negative nitrogen end of another polar molecule is called a covalent bond. Hydrogen bonds are weak, yet stable. That is, they interact unless disturbed with an increase in temperature or an interaction with other chemicals. A water molecule, for example, tends to form hydrogen bonds with other water molecules. Above 0 degrees, however, body temperature is 37, increased molecular movement breaks the hydrogen bonds in water as a liquid. But it is hydrogen bonds. The bonds between water molecules are hydrogen bonds, okay? So they're hydrogen bonds. Good. Good job. Okay. What is the most abundant compound in the human body, accounting for 2/3 of the weight of the body? Water, protein molecules, sodium chloride, or calcium? Water is the most abundant compound in living material and accounts for about 2/3 of an adult human. It is the major component of blood and other body fluids. Mm-hmm. The two components of substances that are blank separate when they are placed in water. So what kind of bond causes compounds to separate in water? Covalently bonded substances, ionically bonded, or radioactive? Covalently bound, ionically bound, or radioactive?
Be ionically bond.
Ionic. That's correct. Ionically bound. Mm-hmm. Good job. Next question. Indicate the four, so this time we're choosing four. I indicate the four types of organic compounds important in cell structure and function. Acids, nucleic acids, salts, proteins, bases, carbohydrates, and lipids. We need four of those that are important in cell structure and function. Okay, so important groups of organic chemicals include these macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So those are the four: carbohydrates, lipids, proteins, and nucleic acids. Okay. Uh-huh. Uh-huh. And lipids. Uh-huh. Right. Compounds that release hydrogen ions when they dissociate in water are classified as what? So what do we call those things that release hydrogen?
Would it be covalent bonds?
This time we're looking for, that's the bond, but this time we're looking for the compound itself. What do we call the compounds that do that? So it says, so here you have bonded substances, right? Those are acids. Okay. They're substances that release ions in water are electrolytes. But if you're releasing hydrogen, if you scroll up a bit or scroll down on the page and past the picture, so it says acids are electrolytes that release hydrogen ions in water. So they're acids. Okay. They're acids. Yeah. The primary function of the class of organic compounds called blank is to provide fuel for cellular functions. So which of the choices provides fuel for cellular functions? Proteins, lipids, nucleic acids, or carbohydrates? Okay. Carbohydrates provide much of the energy that cells require. They supply materials to build certain cell structures and are often stored as reserve energy supplies. So carbohydrates are the energy machine. Okay, good. Next. Compounds called blank bind hydrogen ions or release hydrogen ions, such as hydroxide, that combine with hydrogen. That's bases, I think. Okay, that's our, okay. Then they go down, they talk about pH down here, right? but it talks about how bases, okay, this is the, right, but bases are the ones that release hydrogen. That's kind of in the middle under, go above the practice. Okay, so if you look here, a value called pH is a measure of hydrogen ion concentration. pH stands for potential hydrogenation, the potential for the system to accept hydrogen ions. The scale ranges from 0 to 14. Okay. A solution with fewer hydrogen ions than hydroxide has a pH greater than 7 and is basic. Yeah. So if you release the compounds that bind hydrogen, then it's a base. So I would say it's a base. Compounds called bases bind hydrogen ions. Yeah. Organic compounds, by definition, contain carbon and blank, are often built of long carbon chains or ring structures. So if it's an organic compound, what do I need besides carbon? Carbon and what? Compounds that include carbon and hydrogen atoms are called organic compounds. Carbon and hydrogen are organic compounds. Hydrogen, good. Monosaccharides are also referred to as amino acids, starches, salts, or simple sugars. Another name for monosaccharide is what? Okay, the carbohydrates. Okay, the number of carbon atoms in a carbohydrate molecule varies with the type of carbohydrate. Among the smallest carbohydrates are sugars. Sugars with 5 carbon atoms, pentoses, or 6 carbon atoms, hextoses, are examples of simple sugars or monosaccharides. So another name for monosaccharide is a simple sugar. A monosaccharide is a simple sugar. Okay. Okay, let's see what else we have. The major component of blood and body fluids is lipids, proteins, water, or carbohydrate.
Would it be water?
I believe so, but check the book. Water is the, okay, but let's look at blood and substances which are, okay, keep going. I just need to keep reading. I need, yeah. A substance that, hold on. I need to see what's most common in the bloodstream. But I think these are the most common. Okay. That has to do with, then these are about carbohydrates and okay, that's further down, but it doesn't say anything about blood so far in here. This is about carbohydrates. We're not reading about carbs right now. We're reading about blood. Lipids are substances that generally are insoluble in water, but are triglycerides are used primarily to store energy. The binding blocks are fatty acids and glycerol. Okay. Each, okay. This is about fats, phospholipids. Let me see if I can. I don't see anything about blood in here. Proteins have a variety. Some proteins combine with carbohydrates, allowing cells to respond. Proteins called antibody metabolism could not occur fast enough if, okay, that's how enzymes work. Okay. Okay. Like carbohydrates and lipids, the building blocks of proteins are amino acids. Each amino acid has an amino group, etc. Okay, but that's still not about. Let's scroll down. This is talking about the different types of amino acids there are. And tertiary structures. Um, this is a protein. This is denatured. This is nucleic acids. Nucleic acids form protein synthesis. Let me see if I can see if it says. Mm, I think it's probably water. I think you're right. Let's try it and see. I don't see anything else that it could be. Let's see. Okay, let's continue. What class of organic compounds has the chemical formula of CH2O with twice the number of hydrogens as there are oxygen atoms in each molecule? CH2O, the class of organic compounds. Okay, so that is, let me see. Let me look it up. So So it's got twice as many hydrogen with twice as many hydrogen as carbon. That's a sugar, I think. If sugar's on your list. Unless they're looking for, okay, um, carbohydrates. That's a that's a carbohydrate with twice the number of hydrogens as carbons would be carbohydrates. Like glucose, for example, if it has 6 carbons, it has 12 hydrogens. That's carbohydrates. I'm pretty sure that that's what they want. Yeah. Proteins also contain nitrogen. So, what are the four main groups of organic chemicals in the body? Organic chemicals. They asked you this before, but they had you choose it. Now they're having you choose the group.
Be this one?
Lipids, yes, I believe so. Mm-hmm. Yeah, good. Nice. Okay, next question. Which is a description of a monosaccharide? Several sugar units covalently bonded, a simple sugar unit, or two sugar units covalently bonded. Monosaccharide. Remember, those are the simple sugars, right?
Would it be this one?
I think so, yes. A single sugar unit. Mhm. Good. Next question. What is the major function of carbohydrates in the cells of the body? To provide a source of energy, to act as catalysts, to provide major structural components, or to provide insulation. What do carbohydrates do? Okay, so it says carbohydrates provide much of the energy that cells require. They supply materials to build certain cell structures and are often stored as reserve energy supplies. Carbohydrate molecules consist of atoms of carbon, hydrogen, and oxygen. These molecules usually have twice as many hydrogen as oxygen atoms, the same ratio of hydrogen to oxygen as in water molecules. This ratio is easy to see in the molecular formula of glucose, which that was a question before. So carbohydrates provide energy sources. That's what carbohydrates are for, is energy. So the major function of carbohydrates is to provide an energy source. Yep, good. Carbohydrate molecules consist, okay, we just read it a minute ago. Carbohydrate molecules consist of what three types of atoms? Okay, so here it is. Carbohydrate molecules. I'm reading the middle paragraph. Carbohydrate molecules consist of atoms of carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen. So carbon, hydrogen, oxygen. Very good. Bases do what? Okay, we need two answers for this, okay? So bases are electrolytes. The middle one. And bases release ions in water that bond hydrogens. Because we got that one wrong before. Okay, so that's that part. A carbohydrate formed from two single sugars bonded together is called a what? Okay, scroll down. You need to find the two sugars. That's a monosaccharide. That's a single sugar. When it has two, it's called it's right here. It's right here. A number of simple sugar molecules may be linked to form molecules of different sizes. Some carbohydrates, such as sucrose and lactose, are double sugars or disaccharides. Here's your word in the blue-green color. Disaccharides, whose molecules each consist of two simple sugar building blocks. Disaccharides. I think there's only one S. D-I-S-A. I don't remember how to spell it though. So go back and check the spelling. But yeah, that's the one. So it's DisaccH. So it's one S and two C's, and then Harides. So it's DISAC. C's. Okay. Yeah, that looks right. That looks right. Good job. Okay, next question. Glucose is an example of a small carbohydrate molecule or blank. Glucose is an example of the number. Okay, I would say a sugar. Glucose is a sugar, small carbohydrate molecule. A sugar. Okay. The group of organic substances that are insoluble in water are the what? So if they don't dissolve in water, but they are organic, what do we call them? Okay. So, okay. Uh, okay, that's not it. The sugars dissolve in water, right? Lipids are organic substances that are generally insoluble in water, but soluble in certain organic substances such as ether and chloroform. Lipids include a variety of compounds, including triglycerides, phospholipids, and steroids that have vital functions in cells. So lipids are organic substances that are generally insoluble in water. Lipids. Which chemical formula would represent carbohydrates? Well, if you have twice as many hydrogens as carbons, then it's a carbohydrate, most likely. So the first one is glucose. So that would be a carb, C6H12O6. That's a carb. How about the other ones? Okay. So carbohydrate molecules consist of carbon, hydrogen, and oxygen. These molecules usually have twice as many hydrogen as oxygen atoms. Here's glucose right here. So glucose is on your list. Okay. What else? Do they have any other chemical formulas for anything else? Let's look. Some glucose molecules, C6H12O6. A number of simple sugar molecules have disaccharides. Okay, scroll down so I can see the chemical formula for it. Okay, scroll down more. It's not giving me a formula. It's just telling me that they're, yeah. So I think go to the question because that's all it says. I would think the first one for sure, C6H12O6, and probably the third one, C5H10O5. I don't know what that's the formula for, but it has twice as many hydrogens as carbon. So let's try that and see. Okay, yeah. Does it tell us which It just tells us, okay, that's the correct answer. Okay, but it doesn't say, okay, because there's twice as many hydrogens. So here on the first one, there's 6 carbons. 6 times 2, there's 12 hydrogens. On the third one, there's 5 carbons, and 5 times 2 is 10 hydrogens. So that's why it got that. Okay, what else? The 3 fatty acids bonded to a glycerol molecule form what type of organic molecule? 3 fatty acids plus glycerol. Dipeptide, triglyceride, polysaccharide, or polypeptide. Okay, but that's not carbs. We know that, so we can scroll past the carbs. I think it's going to be a triglyceride, but look at the triglyceride section. Yeah, so here, triglycerides are composed of carbon, hydrogen, and oxygen atoms. However, triglycerides have a much smaller proportion of oxygen atoms than do carbohydrates. The formula for the triglyceride tristerin, C57H110 oxygen 6, illustrates the characteristic proportions. The building blocks of triglycerides are fatty acids and glycerol. Each glycerol bonds with 3 fatty acid molecules to form a single triglyceride. So the way you get a triglyceride is you build a molecule with 3 fatty acids and glycerol. That's why it's a tri molecule. 3 fatty acids, 3 triglycerol. So 3 fatty acids and triglyceride. Okay. So it's a triglyceride molecule. Okay, next question. Simple sugars include monosaccharides, disaccharides, polysaccharides. Monosaccharides. Good job. Next question. Okay. Okay, so the entire molecule shown is called a free fatty acid, a saturated triglyceride, an unsaturated triglyceride, or a glycerol. Okay, so we have to look back at the book to see what we're looking at. But it looks like we're looking at a bunch of, it's going to be with the triglyceride. So let's keep going. Has to be with the diagrams of the triglycerides. That's we're on the carbs. Yeah. Coming. Looks like it's in the, okay, what is this a picture of? The building blocks of triglycerides are, okay, so we're looking here at saturated fat. B, we're looking at the unsaturated fat with a fatty acid portion, okay? So we're looking at an unsaturated fat when they break off like that. And what else do we have down below? We have something else or no? No, okay, so if okay, so let's take a look at the question again. I would say it's an unsaturated triglyceride because this is broken off. If it wasn't broken here, if they were all straight, then it would be saturated. But I would say it's an unsaturated triglyceride. I think. Yeah. Okay. A carbohydrate contains atoms of blank, carbon, and hydrogen. So if they're carbohydrates, they contain three things. They gave you carbon and hydrogen. What's the other one? Okay, carbohydrates. If you look in the middle paragraph, the second one, carbohydrate molecules consist of atoms of what? You see it?
Carbon, hydrogen, and oxygen.
Carbon, hydrogen, and oxygen. Okay, go back to the question. Now, they gave you carbon and hydrogen. So what's the one that's missing?
Oxygen.
Oxygen. So oxygen goes there. They gave it to you in a different order. That's all. Oxygen. Very good. Yeah. A lipid molecule consisting of blank fatty acids and blank phosphate groups bonded to a glycerol is called a phospholipid. So how many fatty acids and how many phosphate groups do you need to get a phospholipid? Okay, well, we got to look it up and see, right? We got to read about the phospholipids. So, okay, we got to scroll past the carbs because we're on the lipid part. Okay, now we're looking at phospholipids, not fatty acids. So keep going. Keep going. We already answered a question about that. I need phospholipids. Okay, so, okay, now look at the yellow. A phospholipid molecule is similar to a triglyceride molecule in that it consists of a glycerol portion and fatty acid chain. A phospholipid, however, has only two fatty acid chains. In place of the third is a portion that includes a phosphate group. The phosphate portion is soluble in water, hydrophilic, and forms the head of the molecule, whereas the fatty acid portion is hydrophobic and forms the tail. So you have two fatty acids and one phosphate. Two fatty acids and one phosphate form a phospholipid. Okay, so now the next thing says, what does it say? It says, what is the definition of a disaccharide? A disaccharide. Simple sugar unit, more than two sugar units bonded together, or two sugar units bonded together. What is a disaccharide?
Would it be two sugar units bonded to?
That's two. That's very good. Mm-hmm. And disaccharide is two sugar units. Right. Okay. What is the next thing? A blank is a lipid that consists of a phosphate head that dissolves in water and two fatty acid tails that don't dissolve. Okay, what was the word that we just used a few minutes ago? That had two phosphate chains and a I'm sorry, two fatty acids and a phosphate. What do we call it? Do you remember? We literally just had it 2 questions ago. You remember what it's called? You're closer. It's closer. There it is. There's the word. A phospholipid, see the green or the blue-green? A phospholipid molecule consists of a glycerol portion and fatty acid chains. A phospholipid has only two fatty acid chains and a phosphate. That's a phospholipid. Oh, nice. How do lipids differ from other organic compounds? So how are lipids different? They dissociate when added to water. They are insoluble in water, or they contain hydrogen. How are they different? How are lipids different?
Would it be they contain carbon and hydrogen?
No, that's not how they're different because sugars contain carbon and hydrogen too. Okay. Lipids don't dissolve in water, they're insoluble in water. We're looking for how they're different. And sugars also have carbon and hydrogen. They just have, the lipids have less oxygen, but they don't dissolve in water. That's how they're different. Okay. Right. Lipid molecules that include connected rings of carbon atoms are called what? Okay, I don't know that we've seen this yet. Lipid molecules that include connected rings of carbon atoms are. Okay, so let's go to lipids. Okay, now we're looking for connected rings of carbon atoms. So we got to keep going down. Okay. The glycerol portion of all fatty acids differ in the lengths of their carbon chains, but most have an even number of carbon atoms. In some molecules, the carbon atoms all join. This type of fatty acid is saturated. Other fatty acids are bound to the maximum number of hydrogen. Fatty acids with double bonds are unsaturated. Okay. Keep going. A phospholipid has, okay. Keep going. Steroid molecules are complex structures that include 4 connected rings of carbon atoms. So, steroids. Steroids have connected rings of carbon atoms. One of the more important steroids is cholesterol. So they have 4 connected rings of carbon. Those are steroids. Triglycerides are made from cholesterol, glycerol, or glycogen. Okay, we need to go down to triglycerides. Now, triglycerides supply more than, okay, they're composed of carbon, hydrogen, and oxygen. Okay. The formula for okay. The building blocks of triglycerides are fatty acids and glycerol. Each glycerol molecule bonds with 3 fatty acid molecules. So triglycerides are not glycogen, glycerol. Glycerol. Fatty acids and glycerol. Okay, so 3 examples of lipids. 3 examples of lipids.
Would it be fats?
Fats, that's one. I would say triglycerides. And probably the last one is steroids. Okay, now this one is the broken off chain. Remember, so it's a fat. So remember which one this was? If you don't, you can look back. It's in the picture with the oils that we had. So if you scroll down, okay, look, see the one that's broken? Look at the caption. It says it's olive oil and it says that it's unsaturated. So when it's broken like this, it's unsaturated. If you scroll up, you see how this one's not broken? This one has all the fatty acid portions. That's that's a saturated fat. So the one below, the one that you have on your quiz, is the olive oil, which is the unsaturated, because it's broken here. Unsaturated. Okay. Okay, so the 4 functions of proteins. So 4 of these 6 answers are correct. What do proteins do? Four function. Go ahead.
Would we provide structure?
Um, I don't know about that one, but I know they detect and destroy foreign substances. I know that one. And I believe they carry genetic information because DNA technically is a protein. I would say they function as receptors. Let's see if I'm right. They function as receptors on cell surfaces and they form, I don't know if they form enzymes or yeah, we're going to have to look this up because I don't know about the fourth one. Let's go to proteins. That's carbs. We need proteins. That's lipids. We've been in lipids long enough. Let's go to proteins. Okay. Proteins have a variety of function in the body. Many are structural materials, energy sources, or hormones. Some combine with carbohydrates, forming glycoproteins, and function as receptors on cell surfaces. Okay, so we got that. So they function as cell receptors. They serve as structural materials, and they detect and destroy foreign substances in the body. And then metabolism could not occur fast enough to support life were it not for proteins called enzymes, which catalyze specific chemical reactions. Enzymes are discussed in more detail. So, okay, so let's see. So they function as receptors. No, the second one is not correct. They don't carry genetic information. They form enzymes. They detect and destroy foreign substances, and they provide structure. I think those are the 4 that we read. Let's try that and see. We got it. Yeah, so you were right about providing structure. So they provide structure, they function as receptors on cell surfaces, they form enzymes, and they detect and destroy foreign substances. A molecule derived from a triglyceride, so they come from triglycerides, but with a phosphate group in place of one fatty acid, that's a phospholipid. They're asking you about phospholipids again, but they have it fill in the blank this time. Phospholipid. Mm-hmm. Good job. Amino acids are the units of constructing larger molecules called what? So amino acids build what? No, that's not carbs. It's should be under protein. Protein. Um, so if you look down under the blue, it says like carbohydrates and lipids, proteins are composed of atoms of carbon, hydrogen, and oxygen. In addition, all proteins contain nitrogen atoms and some sulfur atoms. The building blocks of proteins are amino acid. Each amino acid has an amino group at one end and a carboxyl group at the other. So the building blocks of proteins are amino acids. So, if you go to your question, it says amino acids are the units of constructing larger molecules called proteins. Protein. Okay. Phospholipids have head and tail regions. Have as head and tail regions. Okay. So two of these answers are going to fit. I think it's the hydrophilic head, which is the first one, and the hydrophobic tail, which is the last one. Yeah, that's what we read. So the head likes water, so it's inside the cell. The hydrophobic tail's outside because it doesn't like that. Hydrophilic head, hydrophobic tail. Okay. Name the three groups that are part of the structure of an amino acid. All three are bound to a central carbon. An R group, a side chain, a glycerol group, a carboxyl group, a phosphate group, an amino group, and a nitrogenous base. So think about chemically what makes an amino acid. Okay, you have to scroll down some more. Find the amino acid. Okay, so keep going. Keep going. That's still carbs. Keep going. Okay, so keep going. Okay, look at the last paragraph. The building blocks of proteins are amino acids. Each amino acid has an amino group, that's the NH2 group, and a carboxyl group, that's COOH. So they have an amino group, they have a carboxyl group. Each amino acid also has a side chain or R group that may be thought of as the rest of the molecule. The composition of the R group distinguishes one type of amino acid from another. So an amino group, a carboxyl group, and a side chain. So amino group, carboxyl group, and a side chain, the R group. What is the classification of lipid molecules that consists of connected rings of carbon atoms? So what kind of lipids had those rings of carbons? Phospholipids, steroids, triglycerides, or glycerol? Okay, so let's go to our lipids. Let's look for the rings of carbon atoms. Tell me what kind of lipid it is. Okay. What is the class of it? Okay, so what is the class that have rings? You remember? Yeah, steroids. Good job. Good job. Steroids have rings of carbon. Okay. What type of protein structure is enabled by the given diagram. In other words, is that a primary, a secondary, a tertiary, or a quaternary structure? Well, let's look at the structure of the protein and see what we can find out. Okay, this time we're going to proteins. Okay, so let's go to proteins and let's look for the picture of the protein. We're still on lipids. Okay, we're getting warmer. Keep going. We need to find the picture of our, okay, there's our amino acids. That's what they look like. Okay, I think we're getting warmer right here. So which picture looks like ours?
The left one.
Right, and it says it's a secondary structure. The polypeptide chain of a protein molecule is often either pleated, mean folded, like pleated, like those folds that you see, or twisted to form a coil. Dotted lines represent hydrogen bonds. Our groups are highlighted in orange. Examples of individual amino acids are highlighted in yellow. So the pleated structure is secondary. The pleated or coiled structure is a secondary structure. So it's secondary. Okay, cool. Steroids and steroids and phospholipids are examples of organic compounds called what? What's another name for steroids and phospholipids? So we have to scroll past the carbs. Okay, a phospholipid, steroids, and okay, keep going. A steroid is a complex structure that includes 4 rings of carbon atoms. One of the more, okay. So now what are they examples of? So what are they under? So if we go up the page, it should tell us, unless there's another name that it wants, because I don't think it does anything after that. Okay, so triglycerides there, steroids are there. What are the examples of? I think they're examples of lipids. Lipids are a variety of compounds, triglycerides, phospholipids, and steroids that have vital functions in cells. The most abundant lipids are the triglycerides. I think they're examples of lipids. Triglycerides, phospholipids, and so I think you would just write lipids. Lipids. I think, yeah. What term describes what happens to the protein molecules within an egg white that causes the change in its appearance when it's heated? We read this, but not for a while. This has been a while ago that we read it. That's denaturation. It's the middle term, the middle word. They become denatured. Just like radiation does the same thing to cells. Okay. What class of organic molecule serves a major structural function to the body, as well as functioning as catalysts, cell receptors, and hormones? So what structure does all four of those things? Structural functions, catalyst, cell receptor, and hormone. Okay. Well, carbohydrates serve as energy sources, right? So that's not it. It's not carbohydrates. Lipids are the ones that serve as long-term energy sources. And then we have proteins after that. Proteins serve a variety of functions in the body. Many serve as structural materials, energy sources, or hormones. So proteins serve those functions. Proteins. Okay. Next question. What is meant by the term denaturation of a protein? Loss of three-dimensional shape and therefore function, a change in the amino acid sequence resulting in a change in the type of protein, breaking apart a protein into its individual amino acid subunits, or binding together of two or more polypeptide chains to form a new protein. What happens when you denature a protein? Okay, so go to proteins. Okay, go here. Okay, keep going because we haven't gotten to the denatured part yet. This is your types of structures. When hydrogen bonds in a protein break as a result of exposure to excessive heat, radiation, electricity, pH changes, or certain chemicals, a protein's unique shape may be changed dramatically or denatured. So denaturation is a change in the shape of the protein that comes from the breaking of hydrogen bonds. Okay. So go to the question. So denaturation is a loss of three-dimensional shape. Yes. What are the building blocks that form proteins? Okay, we had this question. What are proteins made from?
Would it be fatty acids?
Not fatty acids, amino acids. Amino acids make proteins. Fatty acids and glycerol make triglycerides, which are lipids. Okay. Don't confuse those two. Amino acids are the building blocks of protein. Triglycerides, which are lipids, are made from fatty acids and glycerol. Okay. Okay, let's see what we got here. Genes comprising DNA carry information about the structure of steroids, nucleotides, carbohydrates, or protein. Okay, this is a different concept. I think we've seen this before. The structure of, let's look it up. Okay, keep going. It's not carbs. It's not anything we've read so far. We have to go into the structure of, okay. This is a type of protein. Keep going. This is, yeah, that's not DNA. We're looking for the double helix problem here. Okay, let's see. There's my protein. Hold on. Nucleic acids. Okay, wait a second. For most proteins, the conformation, which determines its function, is always the same for a given amino acid or primary structure. Thus, the amino acid sequence that determines the role of a protein in the body. Genes made of the nucleic acid DNA contain information for the amino acid sequences of all the body proteins that a cell can decode. Nucleic acids form genes and take part in protein synthesis. These molecules are generally very large. They include atoms of carbon, hydrogen, oxygen, nitrogen, and phosphorus, which form building blocks called nucleotides. Each nucleotide consists of a 5-carbon sugar, ribose or deoxyribose, a phosphate group, and several nitrogenous bases found in nucleic acids. Okay, so genes contain the information for the amino acid sequence. Okay, so let's go back to the question. So genes carry structures about it's not steroids and it's not carbohydrates. I would say protein, because nucleotides come later. I would say protein is the answer they want. Yeah. Relate to protein structures. What is the name of the type of organic molecule consisting of an amino group, a carboxyl group, and an R group? That's a that's an amino acid, I think. Is that an amino acid? I think it is. Yeah, because phospholipids is phosphate and fatty acid. Triglycerides are 3 fatty acids. So a nucleotide is the 5 carbon with a phosphate and nitrogen. So the amino acid is the amino acids build the proteins that have the carboxyl, the R group, and the amino group. Okay, now list 2 characteristics of nucleic acids. 2 of these are, so we have to go back to nucleic acid and read about that. Okay, so nucleic acids form genes and take part in protein synthesis. These molecules are very large. They include atoms of carbon, hydrogen, oxygen, nitrogen, and phosphorus, which form building blocks called nucleotides. Each nucleotide consists of a 5-carbon sugar, a phosphate group, and several nitrogenous. Okay. Is there anything else about nucleotides? Under the video, maybe? Okay. Nucleic acids are of 2 types. One type, RNA, ribonucleic acid, is composed of molecules whose nucleotides have ribose. Most RNA molecules are single-stranded polynucleotide chains, but they can fold into shapes that enable them to interact with DNA. The second type of nucleic acid, DNA, deoxyribonucleic acid, has deoxyribose and forms a double polynucleotide chain. The 2 chains are held together by hydrogen bond. Adenine always pairs with thymine. Cytosine always pairs with guanine. Okay, so let's go back to the question. Right now, they're just asking us about what, so nucleic acids form genes and they take part in protein synthesis. So the first one and the last one. They form genes and they take part in protein synthesis. Okay. The blank sequence of a protein corresponds to individual amino acids. Okay, so now we're back to proteins. They're asking us more about protein. So we need to go back and look at protein. the blank group of a protein. Okay, so that's the functions. Okay, like carbohydrates, lipids and proteins are core. Okay, in addition, all proteins contain nitrogen atoms. Amino acids are the building blocks. Each amino acid has an amino group at one end and a carboxyl group at the other. Each amino acid also has a side chain or R group that may be thought of as the rest of the molecule. The composition of the R group distinguishes one type of amino acid from another. Okay. Okay, keep going. 20 different types of amino acids make up the proteins of most living organisms. The amino acids vary in length. Proteins have several layers of structure. Hmm. Okay. What else do they have about proteins? Okay, the structures, tertiary structure, that's not what it's asking us. The levels of protein structure. Okay, so here, the levels of protein structure determine the overall three-dimensional conformation, which is vital to the protein's function. So the levels of protein structure determine um, the conformation. Okay, proteins with more than one polypeptide chain, the constituent polypeptides are connected. Hemoglobin is a quaternary protein. Okay, keep going. I'm going to see if they say anything else. The levels of protein structure, hydrogen bonding, covalent bonding. The conformation of a protein determines its function. So go back to the question. The blank protein. Oh, okay, that's not what the blank sequence of a protein. Okay, hold on. I'll have to look this one up because I didn't get it. The blank structure of a protein. The blank protein. And what is the corresponds to corresponds okay hold on. Corresponds to individual amino acid. Um the I'm not sure if it's looking for the the peptide structure, maybe? I'm not sure what it's looking for, honestly. Um, mm, I don't know if it's looking for a peptide or conformation here, but because I didn't see anything in particular in the reading. So, but it's the way the peptide bonds are put together. You can try conformation because I saw that, but I'm not sure if it's the word that it wants. Try that and see. I don't know what it wanted. Oh, primary. Okay, the primary sequence of a protein is corresponds to the okay. So basically, when we're talking about the primary level, we're talking about what amino acids. Okay, I understand what the question is asking now. I didn't understand it before, but the primary structure is what amino acids we have. Okay. Okay. I'm sure it'll come back. Let's do. When proteins are exposed to unusual conditions of heat or temperature, the protein may become denatured. They become denatured or altered irreversibly. Denatured. 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 Okay. Nucleotides are used to make very large molecules known as nucleic acids. So acids goes in the box. Nucleic acid. Okay. All right. Next question. When heat or pH or radiation alters the unique shape of a protein, the term is denaturation. So denaturation. Denaturation. It's asking you the same question in different ways, basically. It's asking you in different ways about denatured proteins. Okay, next question. A nucleotide. Okay, so here we're talking about DNA again. A nucleotide contains a nitrogenous base, a phosphate group, and a 5-carbon sugar. And those are the three. Yeah. Okay. What is the name for the DNA sequence that determines the amino acid sequences of proteins? The name for the DNA sequences that determine the amino acids for proteins. Okay, you can go back and look this one up. The DNA sequences that determine the amino acid. Okay, so we have to go to okay. Nucleic acids form genes in protein synthesis. Okay, we just answered this question about the structure. Okay, and then it says nucleic acids form two types. One type is composed of molecules whose nucleotides have ribose. The second type has deoxyribose. These chains are held together by hydrogen bonds. Okay, scroll up. I think we missed something just before we hit the video. Okay. A nucleic acid molecule consists of a chain of many nucleotides. There are 5 different nitrogenous, okay. Okay, keep going. Genes made up the nucleotides that can, okay, let's go to the question and see if, um, I would say genes. Genes is the name for the DNA sequences that determine the amino acids of proteins. I think that's genes. Maybe. Yeah. What type of nucleic acid contains the information needed? What type of what type of nucleic acid stores within the cell nucleus the information needed to manufacture protein molecules? I would say that's DNA because they're within the nucleus of the cell. I don't know what else we would call it because, yeah, okay. RNA uses genetic information to, yeah, because you need DNA first before you can produce RNA, okay? Nucleic acids are most directly involved in lipid metabolism, storage of energy, protein synthesis. I would say protein synthesis because that's what we're talking about is synthesizing or putting together proteins would be my guess. Okay, now what kind of structure is this one? This one is in the book. Which kind of structure is it? Okay, what does it say? Tertiary. It's tertiary structure. So it's tertiary. Good job. All right, next question. A nucleic acid molecule is a chain of many subunits called what? Nucleic acid molecule. So what is a nucleic acid molecule made from? is what it's asking. So you have to go to nucleic acid. And, okay. Okay, so nucleic acids form genes and take part in protein synthesis. These molecules are generally large. They include atoms of carbon, hydrogen, etc., which form the building blocks called nucleotides. So, nucleotides are made from nucleic acid. Okay. So let's go back and so a nucleic acid molecule is a chain of many subunits called what?
Would it be nucleotide?
I think so. Yeah. I think it's nucleotides. If I understood what the question was asking. Nucleotide. If I understood what it was, yeah. The organic building blocks called blank consists of one 5-carbon sugar, one phosphate group. Okay, that's nucleotide again. The organic building blocks called nucleotides. Just asking you a different way. Okay. Molecules of blank store information needed to manufacture protein molecules. I would say DNA. I think it's probably DNA. It's a little bit hard to tell there, but I think it's that. I'm not sure, but yeah. Because they're talking about the genetic information. And I think we're done. I think we're finished. Wow. Okay. That took a lot. What time is it? Almost 11:30. Okay. All right. So that's the end of that one. So I guess tomorrow we'll work on the next unit, or do you have something else that you need to work on tomorrow?
Yes, I think work on like the next unit and also like some notes for like the lab.
Okay. All righty. So I'll see you at 9 tomorrow.
Yes.
All right, Daniela. Take care, okay?
Okay, thank you.
You're
Bye.
welcome. Bye.